I think most of you know that the T32 has been around a long time here at the university. It's housed in the Masonic Cancer Center, and it was renewed last year for years 46 through 50. And one of the interesting changes that we proposed was that we would have our postdoctoral trainees giving seminars in our seminar series. And so today we have Kiel Tietz from the DAME lab, as well as Ethan Aguilar from the Blazer lab. So how it's going to work is that each trainee will present a shortened version of their research, followed by 5 minutes of questions. And so I will introduce both so that they can just go for it. So Kiel Tietz is a postdoc in the Scott Dame lab. He completed his undergraduate degree at the University of Wisconsin in applied science and with a mathematics minor. He then completed his PhD at Ohio State University in molecular, cellular, and developmental biology. And Kiel is an expert on mRNA processing and how that affects disease and development. And he is becoming an expert on how the androgen receptor variants get created. Ethan is a member of the Bruce Blazer Laboratory. He finished his undergraduate in biochemistry and molecular biology at University of California at Santa Cruz. From there, he went to UC Davis and received his PhD in immunology. And Ethan is an expert on T-Regs, or regulatory T cells, in the context of graft versus host disease. And they're both going to tell you about their work today, beginning with Kiel. Thanks, guys.
All right, hopefully you guys can see my screen here. So yeah, I'll get going. Let me just. Okay, so I just want to start by, definitely thanking Carol and the Masonic Cancer Center for allowing, me to give a talk today. I think it's a really good resource for postdocs to be able to do this. But I'm, as Carol said, Kiel Tietz. I'm a postdoc in Scott Dame's lab. And I'm going to tell you about my project that's titled AR Alternative Polyadenylation as a Therapeutic Vulnerability in Prostate Cancer. And so I just want to start by emphasizing that prostate cancer is the most commonly diagnosed cancer in men in the United States. This is just a chart from Cancer Statistics 2021. And you can see here prostate cancer right at the top is estimated to be 26% of new cancer cases in males. And in estimated deaths, it's second only to lung and bronchus. So it's an extremely prevalent disease. And as we continue to age and have longer lifespans, it's only going to become a more prevalent disease. And so I just want to also walk you through the typical prostate cancer progression and the treatment that patients would receive. And so on the y-axis here, we have disease progression. On the x-axis is just time. And so, when a typical man starts to develop prostate cancer, the disease progression goes up. And that's when, really surgery or radiation therapy would be done to destroy or remove the tumor. And for majority of the patients, this cures the disease. But unfortunately, for a subset of these patients, the disease progression continues on. And that's when castration is really the frontline defense here. And these are castration sensitive prostate cancer, because you can see the disease progression does decrease in these patients. But again, unfortunately, it's a matter of time until these castration-sensitive prostate cancer will develop a phenotype of castration resistance. And I don't have time to go into it today, but it has been shown that reactivation of the androgen receptor is, in its signaling, is really a critical driver of this castration-resistant phenotype. And so these males, will develop much further disease progression. And that's really when next generation AR therapy comes into play. And that's, really potent anti-AR targets or drugs such as enzalutamide. And so, enzalutamide will be given to these patients and it will help them temporarily. But again, unfortunately, it's only a matter of time until this castration-resistant prostate cancer will develop resistance to even these drugs. And, the disease progression will just start to skyrocket and ultimately lead to the death of the patient. And so in Scott's lab, we're very interested in this castration-resistant phenotype of prostate cancer, specifically the reactivation of AR signaling and better understanding mechanisms that drive this reactivation and druggable targets that could help inhibit this reactivation of AR. And so I also want to just walk you through the androgen receptor. So this is the canonical gene structure. It's an 8 exon gene that gets encoded to the AR transcript, translated to AR protein, which importantly has these 4 domains: the NTD, the N-terminal domain, the DNA binding domain, the hinge region, and the ligand binding domain. And so importantly, the DNA binding domain and the N-terminal domain are really important for downstream activation of target genes. And the ligand binding domain is important for sensing the presence of androgens. And so Scott's lab and work from others have detected that in patient samples, as well as prostate cancer cell lines that we use, there is isoforms of the androgen receptor, these androgen receptor variants. They're actually truncated forms of the androgen receptor. And this is just an example of ARV567ES, which really, as the name denotes, it's a deletion of exon 5 through 7 in these patients. And so, this genomic deletion really results in a frame shift of exon 8 and critically a loss of this ligand binding domain. So you can see this would be detrimental because now you have this variant of this androgen receptor that has intact everything it needs to activate downstream target genes, but it has lost its ability to sense the presence or absence of androgens. And so my project is more focused on the post-transcriptional regulation of the androgen receptor. And so again, Scott's lab has shown, and work from others, that through alternative splicing, as well as more my project, alternative polyadenylation, is used to include these cryptic exons such as CE5, CE3 that reside within this intron 3 of the AR. And so it really ultimately includes intronic sequence into these transcripts, which ultimately has the same fate as what I just showed you. It causes a frame shift and a loss of this ligand binding domain. And so these are just ARV7 and ARV9, both very clinically relevant variants. And ARV7, for instance, is actually used in cases to predict enzalutamide resistance. So really, my project is focused on how alternative polyadenylation is working to generate these androgen receptor variants. And so I know many of you don't study the process of cleavage and polyadenylation, so I just want to spend a little bit of time just emphasizing what this is. And so mRNA contain this canonical polyA signal, which is this AAU, AAA, and it's typically located in the last exon of mRNAs. And really the polyA site is again, typically a CA sequence that's following this polyA signal. And that's really where this cleavage occurs and this polyA tail is added onto the 3' end of this mRNA. And so this polyA tail is critical because it's important for stability, translocation, and also, translation of this mRNA. But, really importantly, this process is critical because it defines the 3' end of the transcript. So you can see if you used a polyA signal more, upstream or downstream, you would get a longer mRNA molecule, which would encode for a longer protein. And I do want to say, while we're very interested in alternative polyadenylation in prostate cancer, this mechanism has been linked to disease and cancers. So there's, just shown here, form of muscular dystrophy has been found to use more proximal polyA sites, so shortening the 3' UTR. There was a paper actually about 2 years ago that came out that showed widespread intronic polyadenylation and chronic lymphocytic leukemia. And this is actually very similar to what we see with the androgen receptor. So this was, suggesting that this mechanism is could be manipulated in multiple cancers. And so I lastly just want to introduce, there are many factors that are involved in 3' end processing. And I'm not going to go through every one of these. I just want to show you that, there is a lot of factors that are known to regulate usage of the polyA site. So there's, 4 subcomplexes. The CPSF complex is something we're very interested in. I'll show you data on, but that subcomplex actually directly interacts with this AAU, AAA. There's the CSTF complex, which is known to bind to downstream sequence elements, and then also the CF1 and CF2 complex that is known to bind to upstream sequence elements. So all of these factors really play a role and have been found to play a role in selection of the polyA site. And work from Sarah Monroe in the Minnesota Supercomputing Institute has also showed clinical significance of specific 3' end processing factors. So as you see here, this is a progression-free survival plot, just looking at TCGA data. And we can see here with CPSF1, a member of that CPSF complex, as well as PCF11, we see that lower expression of both these genes lead to better progression-free survival. And we see the reciprocal is true for CPSF2 and CSTF1, where higher expression actually correlates to better progression-free survival. And Sarah also looked at, importantly, CRPC, so castration-resistant prostate cancer patients, their expression versus primary prostate cancer, and looked at expression of these genes across 4 different data sets. And what we find is, specifically for CPSF1, we see enrichment of this gene across these 4 different data sets in CRPC versus primary prostate cancer, showing clinical significance of specific 3' end processing factors. And so a previous postdoc in the lab, Jamie Van Etten, who was actually also a T32 fellow, had published a paper a few years back that showed that when we did RNA-seq analysis of 5 different patient samples, as well as 22RV1. This is a cell line I'll be showing a lot of data on. It's a ARV positive CRPC cell line that really, regardless of the cryptic exon that's used to generate variants within these patients and the cell line, the RNA-seq reads all really fell off at the very end of this CE3, this cryptic exon 3, right at this polyadenylation alternative polyase site. And just to help visualize that, again, independent of which splice sites used, within these cryptic exons and intron 3 of AR, all of these different variants, the reads fell off right at this alternative polyase site. And so that was, a pretty big flag for us. And Jamie decided that she wanted to lay an antisense oligomer, a morpholino, down directly on that polyase site and see if we can disrupt a generation of variants through alternative polyadenylation. And so that's exactly what she did. She took 22RV1 cells, and this is termed CE3 PAM. It's this morpholino. And you can see here in control, this is 22RV1 cells. When we probe for total AR using an N-terminal domain that detects all isoforms of AR, you can see the androgen receptor variants are these truncated isoforms down below. The full length AR is right here. But when we use this CE3 PAM, you really completely lose expression of ARVs in these 22RV1 cells. And I would say more importantly, actually, we even shift the expression to higher full length AR. And that's quantified here in qPCR data, where we can see a over 2-fold increase of full length AR and a significant reduction of all these different variants. And so, as I said, this is really important because we're not only inhibiting the expression of the variants in this cell line, we're also now driving back expression to full length AR, which could be very important for patients, to help be more sensitive to, say, drugs as enzalutamide. And so what I've done since joining the lab is design more morpholinos, more antisense oligomers, to see is there other sequence elements that are helping influence this generation of androgen receptor variants right next to this alternative polyase signal. And so, this is that CE3 PAM. If we just zoom in right at this very end region of CE3 that Jamie had developed, but what we had developed now is morpholinos that targeted either upstream 50 or upstream 100, and these denote roughly the nucleotides upstream of this site, or downstream 50 or downstream 150. Unfortunately, there's a very repetitive sequence that's just hard to target here. But ultimately, the goal is to see with the diagram I showed you earlier, are upstream and downstream elements also playing a role in regulating usage of this alternative polyase site. And so what I did is I used these morpholinos in these 22RV1 cells, very similar to what Jamie did. And what was really interesting is we saw actually a reciprocal effect to what we see when we lay a morpholino down directly on that alternative polyase site, where when we look at the downstream 50 and an upstream 50 region, we actually now completely lose expression of the full length AR and drive more now expression of these ARVs in this 22RV1 cell line. And specifically with the downstream 50, when you look at V7 or V9, these variants alone, we definitely see an increase in the expression of these variants when we disrupt this downstream 50 sequence. And so that really suggested to Scott and I that some kind of transacting factor could be laying down right on this downstream 50 region to actually negatively regulate usage of this polyadenylation site. And to further confirm that hypothesis, we examined LNCAP cells. So these are ARV negative, androgen sensitive prostate cancer cell lines. And so, as I just said, they're ARV negative. So when I did this treatment with these different morpholinos and we did a normal exposure with the Western blot, you only see the full length AR. However, when we did a longer exposure, you can start to see this non-specific signal, but with the downstream 50, you see this clear doublet here. And then when I probed for V7 specifically, it was clear that we're inducing now expression of an invariant in an otherwise ARV negative cell line, which was a very interesting result, but also solidified the hypothesis that something in this downstream 50 region is really negatively regulating usage of this poly-A site, even in ARV negative cell lines. And that kind of leads into the other aspect that we're very interested in, which what are the 3' end processing factors? What are the transacting factors that regulate usage of this alternative poly-A site in CE3? I just showed you data that we know sequence elements are critical for regulating negatively and positively usage of this site, but really what are the factors that are helping drive this? And so just going back to the patient survival, or sorry, the patient data that we have, this really nominated CPSF1 as a factor we're really interested in, because again, we saw that low expression of CPSF1 correlated with better progression-free survival. And CPSF1 was actually the only gene that stuck out across all 4 data sets that were enriched in CRPC versus primary prostate cancer, suggesting that this gene could be upregulated to drive a more CRPC phenotype in patient data. And so work that Jamie had done, and also some preliminary work of mine, has shown that CPSF1 also can regulate the expression of AR and ARVs. And so when Jamie did just transient knockdown with CPSF1 by siRNA, you can see here that the variants do pretty strongly decrease when you treat with these siRNAs. And when you probe for V7, you see a similar result. And it's not as easy in this Western blot, but we have other data to solidify that it actually does increase AR expression, the full length AR. So this is pretty similar to what we see when we actually laid that morpholino directly on that alternative poly-A site. And what I've shown more recently in some preliminary data is that in these LNCAP 95, so this is now the LNCAP 95 is a cell line derived from the LNCAP cell line that is actually ARV positive now. And it's androgen insensitive. But what you can see here is when I use CPSF1 shRNA, so stable knockdown, we clearly lose expression of ARVs. And then when I probe for V7 again, it is pretty much gone in this LNCAP 95. So we have 2 different cell lines here now, where a knockdown of CPSF1 has a pretty similar result to what we see when we inhibit the direct alternative poly-A sequence itself. And what I also did is just look at endogenous CPSF1 levels. And so this is, again, LNCAP, that ARV negative line, and this is 22RV1, the ARV positive line. And what really stuck out to us is when we just look at endogenous levels of CPSF1, it is dramatically upregulated in 22RV1 versus LNCAP cells. And again, this fits our hypothesis that this factor could be upregulated to help drive a more CRPC phenotype. And I also did look at the function of CPSF1 in just 2-dimensional growth of prostate cancer cells. And so I did, shRNA is a stable knockdown across these 3 different cell lines, the 22RV1, LNCAP 95, as well as LNCAP. And what you see here across the board is when you knock down CPSF1 in these prostate cancer cell lines, if you just compare day 0 to day 6 growth, there's a clear phenotype of inhibiting growth in these prostate cancer cell lines. And specifically, Scott and I are very interested in LNCAP because, again, this is an ARV negative cell line. And so, we're still seeing a clear growth effect. And we're very interested in also mechanisms that could be AR independent, that CPSF1 is regulating prostate cancer cell growth. And so, with that, I just want to conclude what I talked about today. I showed you that, in this cryptic exon 3 of the AR gene, we found that, specific sequence elements can drive the expression of full-length AR and ARVs. Specifically, when we inhibit the alternative poly-A site, we drive stronger full-length expression, lower variant expression. But if we inhibit the flanking sequence, we actually get the reciprocal effect, where we get lower full-length and higher ARVs. And this really solidifies that, there's a lot of interplay going on between these sequence elements that are driving selection of the full-length versus ARVs in these prostate cancer cell lines. I also showed you that we've highlighted of the CPSF complex, CPSF1, to really have clinically relevant data where we see that low CPSF1 expression has higher progression-free survival, and also CPSF1 is enriched across all these data sets in CRPC versus primary prostate cancer. And just lastly, as I showed you previously, when we knock out CPSF1 in prostate cancer cell lines, we see a shift of more like full-length AR, similar to when we inhibit the alternative poly-A site itself. We see a reduction in ARVs and also an overall reduction in cell growth. And so really quickly, I just want to talk about what we're really excited to be doing. We have some ongoing work that I think is definitely the future of the project, which is determining global mRNA targets of CPSF1. This also goes into what I said, where we could develop or understand a AR independent mechanism of CPSF1 in prostate cancer. But we're interested in identifying global targets in any differential binding between, say, androgen responsive versus androgen insensitive prostate cancer. And so, I don't want to go into the details of this, but we are using this technique called E-clip. It's an enhanced clip where you get very good detection of RNA binding of proteins that you're interested in. And so again, what we're going to do is look for CPSF1 in cell lines and patient data or patient tissue to see, can we see global targets of CPSF1? And again, is there differences in androgen insensitive versus androgen sensitive tissues and cell lines? And then secondly, just the last thing I want to talk about is data that I think would couple very well with what I just showed you is determining the global polyadenylation changes when CPSF1 levels are altered. And so again, I don't want to go into the nitty gritty of this, but we're going to be using this poly-A click-seq. It's a click-seq chemistry. And in a nutshell, when you compare it to RNA sequencing, this is the PAC-seq, you get very good coverage on the 3' end, even to single nucleotide resolution to see where the 3' cleavage and polyadenylation is occurring. And so again, we're going to do this in cell lines and patient specimens and see specifically in cell lines, if I knock down CPSF1, do we see now these global changes in alternative polyadenylation? And does that correspond to targets that we've identified by that E-clip method? And so with that, I just want to acknowledge the entire DAME lab. Scott's been a great mentor for this project. And also MSI, the Minnesota Supercomputing Institute. Sarah, did a lot of work helping with analysis, and so is Rebecca and Rendon. And with that, I'll take any questions you guys have.
Wow, Kyle, that was quite beautiful. I'll start with the questions. So, you kind of got to the idea that there's probably other genes that also are susceptible to this kind of regulation at the same time that could maybe synergize or work when AR, when things are resistant. So twofold question. Are there other cancers where this factor is known to be greatly upregulated that come to mind, like breast cancer, and also other steroid receptors? So that particularly the glucocorticoid receptor, which is kind of fascinating. It's one gene, but there's been dozens of splice variants described, and in thousands actually of variants that occur by SNPs. So have you looked at other steroid receptors in prostate cancer that could be regulated this way, or are there other cancers that are like obviously maybe a place to look for this factor?
No, that's a really good point. And it kind of goes back to my intro slide where, so CPSF1 itself, I'm not sure if that specifically has been linked to other cancers, but PCF11, another factor that I showed early on that actually had clinical relevance in prostate cancer, which is also a 3' end factor, has been found in neuroblastoma to have a role as well. And so, off the top of my head, I'm not sure. And I don't think we've looked in breast cancer about expression patterns of CPSF1. And really, to my knowledge, we're the first one besides Jamie's data to really look at CPSF1 in prostate cancer. So, it does kind of seem like it's a newer connection to the cancer field, but I think it's, yeah. And also to your second point, I have not looked at the glucocorticoid receptor, but it would be interesting to see. And that's why, I think right now, literally what we're doing is, I want to get global data sets so that it'll be really great to look at, like we're really not trying to hone our focus only on AR. I actually think it would be really exciting to find AR independent pathway that CPSF1 is regulating these cancers as well. So
sure. I mean, it seems like most of the field is focused on AR, and as much as we love AR maybe doesn't do everything, right?
No, I know. It actually would be probably more novel if we found an AR independent mechanism, which would be great.
Yeah, well, it's quite beautiful work. And thank you.
Thanks.
That was a gorgeous talk. Absolutely gorgeous. I don't see any other questions. Sandy, do you see anything?
I'm not seeing anything, Carol.
Okay.
Would like one more question? All right.
All right. Well, thank you so
Thanks.
much. That was gorgeous. And now we will hear from Ethan.
All right, hopefully you guys are seeing my slides. So I'm going to talk today about my project in Bruce's lab, where we study just different ways to therapeutically treat graft versus host disease. And my focus has been primarily on regulatory T cells, and specifically looking at metabolic mechanisms or metabolic ways to target these T-Regs to enhance their activity. And so just to give you a brief overview of hematopoietic stem cell transplantation, as you can see, it can be used to treat a variety of different diseases and disorders. And it often remains the only curative option for patients. And again, this list is in no way exhaustive and comprehensive, but it includes things like hematologic malignancies or cancers of the blood, or other hematologic diseases like anemias. It could also be used to treat patients with immunodeficiencies, which are lacking normal immune function, or even lysosomal storage disorders where patients are lacking some key digestive enzyme, et cetera. And even in some cases can be used to treat solid tumors as well. And so just to give you guys a brief overview of the procedure, it starts off with the patient that's going to actually be given the hematopoietic stem cell transplantation. They need to undergo a conditioning regimen that's aimed at eradicating, and in this case of this patient, the leukemia cells. So it's the conditioning regimen eradicates both the leukemia cells as well as the patient's bone marrow, which includes their immune system. And so that leads to the next step where a donor patient, stem cells are harvested from a donor patient and then infused into that donor patient. And then over time, these cells are able to repopulate the hematopoietic system, giving rise to red and white blood cells. Classically, during this phase of repopulation or recovery, this leads to a critical period for opportunistic infections because these patients are currently developing their immune system, and so they don't have means of, or great means of protecting themselves. This also leads to a period for the development of graft versus host disease, which is what I'm focused on primarily. And classically, graft versus host disease that manifests within the first 100 days post-transplant is termed acute, whereas manifestations that happen after the first 100 days of transplant are often termed chronic. We now know that there's considerable overlap between these two diseases. And so it isn't really as clear cut as this, anything within the first 100 days is acute or anything after is chronic. That's not really how we term it anymore. And oftentimes patients that develop acute GVHD will go on to develop chronic GVHD as well. And so again, it's not as simple as 100 days, first 100 days acute, second 100 days chronic. For my purposes, I'm focused on studying chronic graft versus host disease. And this is a leading cause of morbidity and mortality following allogeneic hematopoietic stem cell transplantation. And as you can see, it occurs or it affects anywhere from 30 to 70% of patients. it is characterized, or both acute and chronic graft versus host disease are characterized by or are initiated within these 3 phases of disease. So they all pretty much have the same kind of manifestation. It's just a matter of the cell types and players that are playing a part that's going to lead to either an acute or a chronic disease. But essentially in the first phase of disease, you have tissue damage caused by the conditioning regimen. And this leads to release of pathogen-associated molecular patterns or damage-associated molecular patterns that are able to go on and activate the innate immune system at the site of the tissue damage. Once these innate immune cells are activated, they can go on to initiate the second phase of disease, which is the activation of the adaptive immune system within lymphoid follicles. And so, as I mentioned, the cell types that are activated in this first phase will traffic to lymph nodes, where they will activate the second part, the adaptive immune system. Once that occurs, we can get into the third phase, which is the adaptive immune system that's activated in the second phase, going on to mediate actual tissue damage and disease. And so again, innate immune systems activated in the first phase, they activate the adaptive immune system, and the adaptive immune system is actually the cells that go on and mediate tissue damage. And here on the right, I just have some panels kind of giving you an idea of the kind of issues that these patients can be undergoing. And so panel A and B shows just sclerotic lesions of the skin. Panel C shows chronic GVHD within the gums at the base of the teeth. Panel D is of patient's esophagus. You can see that it's swelled up, which makes it hard for people to swallow. And you could also even have manifestations on the fingernails as well. And so, oftentimes this leads to just a very uncomfortable life for these patients. And so we're interested in how we can treat this. And so just to give you another brief overview of the different cell mediators within that are actually mediating this disease, I actually only have outlined here the adaptive immune cells, the ones that are actually going out and mediating the tissue damage. And so there, as I mentioned, there's the primary cell types. The pathogenesis of the disease is highly dependent on activation of conventional T cells. These are going to be the primary orchestrators that are being activated and actually going on and amplifying and activating other key cell types. Then you have B cells. B cells are highly reliant on these conventional T cells for activation. And once activated, these B cells can go and produce antibody. And these antibodies, once released, are actually what's able to go and mediate tissue damage within the patient. And so we really kind of, if we wanted to limit disease, we would want to limit antibody production by B cells or limit conventional T cell activation, which would hopefully limit B cell activation. A third cell type is the regulatory T cell, which is what I'm interested in. And these cells are able to suppress active immune responses. And so, and also importantly, in some cases of some cases of conventional T cell activation, you can actually have T cells differentiating into regulatory T cells. And so you can have these pro-inflammatory cells in certain contexts turn into suppressive anti-inflammatory T cells. And so here on the right, I just have a simple diagram of a germinal center. And so the germinal center is kind of where all of these cell types come together and meet within lymphoid follicles to actually become activated and start producing antibody. And so once T cells are able to enter, T cells activated outside are able to enter this germinal center and differentiate into T follicular helper cells. They encounter their germinal center B cells, which are also present. They give them signals to become activated. These start producing antibody that it's able to go and mediate tissue damage. And then, as I mentioned, these T-Regs that are able to enter this germinal center differentiate into T follicular regulatory cells, and these cells are actually able to suppress and stop this response. And so, as I mentioned, I've kind of been interested in a metabolic approach to trying to therapeutically treat graft versus host disease. And this whole idea of metabolic regulation of the immune system is actually a very hot area in immunology right now. And the Blazer Lab as a whole has taken a very systems-wide approach to this, where we have projects investigating multiple different metabolic pathways within metabolism to try to treat graft versus host disease. And for the purposes of my project, I'm really just focused on this little tiny red square here, or fatty acid synthesis. And so just to give a brief background on what people have seen when looking at fatty acid synthesis. So there was a prior paper that was interested in investigating the role of fatty acid synthesis during T cell differentiation into a Th17 cell. And so T cells differentiating into Th17 cells are just becoming a pro-inflammatory T cell. And so people were interested in studying what role does fatty acid synthesis contribute to that differentiation. And the way they did this is they targeted these ACC enzymes, or acetyl-CoA carboxylase. There are two isoforms of this enzyme, ACC1 and ACC2. And they are actually the enzyme that catalyzes the first step of fatty acid synthesis. They are able to take acetyl-CoA and catalyze it into malonyl-CoA. ACC1 is present in the cytosol, whereas ACC2 is associated with the outer mitochondrial membrane. But both pretty much mediate the same function where they turn acetyl-CoA into malonyl-CoA. And so this prior paper used either a drug approach to target acetyl-CoA, or they actually had knockouts as well. And they just wanted to look at, with T cell activation in the context of Th17 conditions, what would happen with fatty acid synthesis blockade. And their hypothesis was that because these T cells are going to be undergoing proliferation, activation and proliferation, that fatty acid synthesis would be something that they would be required, or would be required, since dividing cells need new fatty acids to build plasma membranes, et cetera. And so here on the right is just what happens in a normal situation during T cell differentiation. So a naive T cell up on top, once it's activated to become a Th17 cell, it's highly reliant on glycolysis. So it needs a lot of glucose. All the glucose-derived byproducts pretty much feed directly into fatty acid synthesis, which is here on the right. And again, this is in a normal context. But for them, when they actually came in and blocked this step here, this acetyl-CoA to malonyl-CoA with sorpin A on the left, what they found is that this actually led to blocking ACC1 with sorpin A actually led to a dose-dependent reduction in the frequency of Th17 cells, while favoring the induction of FOXP3 expressing T-Reg cells. And so essentially, when you block fatty acid synthesis during T cell activation, you could actually drive T cells towards an anti-inflammatory T-Reg phenotype. And we were actually very interested in extending these findings because we're interested in using T-Regs to try to treat graft versus host disease. And maybe this would be a way where we could both make more T-Regs as well as have better functioning T-Regs. And so to get to the better functioning T-Reg aspects, as you can see here outlined, malonyl-CoA that's formed by that ACC enzyme is actually able to inhibit CPT1A and fatty acid oxidation. And so the whole idea here on the right is if you're actively making fatty acids and utilizing this fatty acid synthesis pathway, you wouldn't want to at the same time be burning the fatty acids that you're making in fatty acid oxidation. It's kind of a one or the other. You're either going to be making fatty acids or you're going to be burning fatty acids this way. And so if you're making fatty acids, that first product that's made during fatty acid synthesis, malonyl-CoA, is able to actually go and inhibit CPT1A, which is the rate limiting step for fatty acid oxidation. Again, you're making fatty acids or you're burning them. And the thing that's important here is T-Regs are actually highly reliant on oxidative phosphorylation for function. And it's been shown that they could actually use fatty acid oxidation very well to drive this increase in OXPHOS and increase in function. And so essentially T-Regs love fatty acid oxidation for their function. And so if we're able to block this step here by using either sorpin A or different drugs, or even knocking out ACC1, could we then drive less formation of malonyl-CoA, less blockage of CPT1A, and increased OXPHOS? Hopefully that makes sense. And so that's essentially my hypothesis, is that inhibiting fatty acid synthesis is going to lead to an increase in OXPHOS. And this increase in OXPHOS will correlate with an increase in suppression. And then we could use these inhibited T-regs to then treat graft-versus-host disease. And we're going to be testing this out a number of different ways. We have ACC1 FOXP3 pre-mice, which harbor T-regs lacking ACC1. And we also have 3 different drugs that we're testing out as well. All are ACC1 and ACC2 inhibitors. They target both isoforms. And so we have sorafenib A, which was used in that original paper. We also have TOFA, as well as ND630. ND630 being like the newest, best one that's currently being tested clinically. And it's been found to be highly specific for mammalian ACCs compared to these other 2 drugs, which are a little less characterized. And so jumping right into it, I first wanted to look and see whether my ACC1 knockout T-regs had any kind of change in oxidative phosphorylation, because as you can remember, my hypothesis was that inhibiting fatty acid synthesis would lead to decreased malonyl-CoA, which should hopefully lead to an increase in fatty acid oxidation, and increase in fatty acid oxidation would lead to an increase in just OXPHOS in general. And so we used a seahorse assay, a Midas stress test, to test this out. And essentially, what we found was that we did see a significant increase in the oxygen or the OXPHOS rate of our knockout T-regs compared to our wild type T-regs. And this was consistent with what we were expecting. And so next I wanted to see whether T-reg suppression was actually affected. And so to do this, we actually co-culture activated T cells in vitro with different ratios of T-regs, and we just measure T cell proliferation. And so essentially we're just looking for the T-regs to inhibit T cell proliferation in a co-culture. And as you can see here on the top is CD4 T cells and on the bottom is CD8s across all ratios. So here's 1 to 1, 1 to 3, 1 to 6, and 1 to 12. And here on the left, just the representatives, all I'm showing is 1 to 3. But you can see across the board, we see a significant increase in vitro suppression by these T cells, consistent with what we were expecting. And then interestingly, when we actually look at these T cells following in vitro activation, we can see upregulation of different, different inhibitory cell surface markers that are commonly found on inhibitory T-regs. So seeing things like LAG3 and TIGIT, which are both inhibitory cell surface markers, as well as CD25, which is a high affinity IL-2 receptor subunit. And T-regs are actually highly reliant on IL-2 for just survival. And so to see this increase is also very interesting and noteworthy. I also looked at just proliferation by Ki67 following activation. And we saw a slight increase, although I don't know that that was much to write home about. But the thing that I was actually interested in was just to see whether there was any effect on viability of the cells following activation. And as you can see, there was really no difference between knock-in and wild type following activation. So in conclusion, targeting fatty acid synthesis enhances their suppression, and this enhanced suppression is correlated with an increase in cell surface inhibitory receptors. And this doesn't come at the expense of cell viability. And so as I had mentioned previously too, we also have these drugs that also target fatty acid synthesis. And so I also wanted to test these out as well. And so just doing the same thing as I did before in a Midas stress test, this time using ND630. Again, consistently and significantly, we can see an increase in oxidative phosphorylation with fatty acid synthesis inhibition. And then looking next at suppression, we also again see consistently a significant increase in suppression. And as I have noted here on the bottom, we've also had very similar results with TOFA and sorafenib A treated T-reg as we do with the ND630. I'm also getting questions. Should I be answering questions during or after?
You can wait till after. These are great questions, but
okay.
I would say let's handle these after. Thanks.
All right, sounds good. I was just seeing them pop up. Anyways, so next I was interested in what the effect of inhibiting fatty acid synthesis is on mTOR signaling. as mTOR can be a master metabolic regulator and is often looked at in these studies. And so interestingly, and kind of consistent with what I was expecting, we see a significant decrease in mTOR signaling. And so looking at the downstream target of mTOR C1 or S6, or phosphorylation of the mTOR C2 targets, AKT and FOXO3A, consistently we see a significant decrease in signaling between these molecules. And then interestingly, downstream of this mTOR signaling complex is DRP1, and DRP1 is a mitochondrial fission promoter. And so essentially when this enzyme is activated, it works to promote mitochondrial fission or breaking up of mitochondria. And so we see a significant decrease in phosphorylation of this as well, indicating maybe mitochondrial fusion might be affected as well. And so working with a group at Oxford who's able to do image mitochondria and look for mitochondrial fusion, we sent them T-Regs as well as TOFA. In this case, we still need to repeat this with our ND630. But interestingly, when we look at mitochondrial fusion in this context, we are able to see a significant decrease in sphericity of mitochondria, which is indicative of an increase in fusion. And so if you actually look at these individual orange mitochondria in the conventional T cells and regular T-Regs, you can see that the mitochondria kind of form these little balls, whereas in the TOFA treated, they're forming more of a longer chain, almost like these little balls have fused together. And so there's been other studies that have shown that fused mitochondria are actually able to facilitate enhanced oxidative phosphorylation. And so that was kind of the idea that we were working on here, where maybe the increase in mitochondrial fusion is what's actually promoting that increased NOx-phos. And so I just wanted to also correlate this with other measures of mitochondrial activity. And so we have these other flow cytometry cell dyes that are actually able to measure, in the case of CellRox, this measures cellular reactive oxygen species. Mitosox measures mitochondrial superoxide. Mitochondrial deep red measures mitochondrial membrane potential, and then myotracker green is an indicator of mitochondrial mass. And as you can see across the board, we see significant increases in all of these, indicative of enhanced mitochondrial activity. And so next, I was interested in asking whether we could limit this enhanced T-Reg activity by targeting OXPHOS at the same time as fatty acid synthesis. And so to do this, we use this IACS inhibitor. And so IACS is a mitochondrial electron transport chain complex 1 inhibitor. And essentially by using this, you should limit OXPHOS because you're targeting one of the electron transport chain complexes which mediate OXPHOS in the mitochondria. And so the whole thought here was that a combination treatment of ND630 with IACS would lead to a loss of that enhanced OXPHOS that I see with just ND630 treatment alone. And that's actually what I saw. So in red, you can see in a normal situation compared to blue, we see that enhanced OXPHOS quantified here on the right. But then when we actually come in purple with the combination treatment, we actually decrease and lose that enhanced OXPHOS that we saw with ND630 treatment. And so next I was interested in what the effect was on T-Reg function or suppression with combination treatment. And consistent with what I would have expected, when we actually do this combination treatment, we actually lose the enhanced T-Reg function that we noted before. And so finally, our last question is, can we use these enhanced T-Regs to treat chronic graft-versus-host disease? And so to do this, our normal setup for this is 2 days before or 3 days before transplant, 2 and 3 days before transplant, mice are administered cyclophosphamide as a conditioning regimen. And then the day before transplant, they actually get hit with 830 gray of total body irradiation as well as a part of the conditioning regimen. And then day 0, they actually get their bone marrow transplantation, which is 10 million bone marrow cells with 70,000 T cells. This 70,000 T cell number is actually very highly titrated down. It's very hard to get these mice to stay as a chronic phenotype, or I should say the opposite. It's very easy to get them to turn to an acute phenotype. And so again, the 70,000 T cell dose is very well titrated down to try to achieve that chronic manifestation. And then every time anytime we're treating chronic graft-versus-host disease mice, we're always starting treatment at day 28. If we started earlier, then there would be questions on whether we're actually affecting engraftment of these cells and not actually affecting disease. And so we wait to day 28 for disease to be established, and then we try to come in and therapeutically treat instead of a prophylactic approach. And so to do this, we adoptively transfer 500,000 wild type or ACC1 knockout T-Regs. And then at day 49 following transplant, we take the mice down and do pulmonary function tests, as well as flow phenotyping to look at those germinal center populations. And so here's just an example of the lung function assessment that we do. And so we intubate our mice onto a FlexiVent system, which is able to measure lung function with these 3 parameters: resistance, elastance, and compliance. Resistance is just a measure of the, resistance is a measure of the constriction of the lungs. Elastance is a measure of the stiffness of the lungs, and compliance is just the reciprocal of elastance, and it's supposed to describe the ease with which the respiratory system is able to function, essentially. But what we're essentially looking at is with resistance and elastance, when we actually have in a chronic situation in gray, we see increases in these parameters as well as a decrease in compliance. And so when we go in and treat these mice, we like to see our treatment groups normalize back to the bone marrow only black bars. And then down on the bottom, we actually are able to look at trichome staining as well as IG deposition within the lungs as well. In this case, this is just trichome staining for collagen. And as you can see around the bronchial and alveolars, there's a lot of inflammation indicative of bronchiolitis obliterans in these mice. And so just moving into the data now with my adoptive transfer. Here in green are my knockout T-Reg transfer group. You can see that these actually had the best disease outcome amongst the 3 chronic GVHD groups, and actually led to a significant decrease in disease burden when compared with the red chronic GVHD group. And so this was suggesting that our ACC1 knockout T-Regs are able to treat chronic graft-versus-host disease as far as lung function at least. And then next, looking at our actual immune phenotyping within the spleen, trying to phenotype again our germinal center populations, you can see that we see a significant decrease in both germinal center B cells as well as TFH cells. Interestingly, and much to my sadness, we didn't see a significant increase or any kind of effect. I mean, if anything, we saw a significant decrease in our follicular regulatory T cells. And so these are those T cells that I'm interested in, the regulatory T cells. And then oftentimes we'll look at the ratio of TFR to TFH, which is usually a little more indicative of a positive disease outcome. And as you can see, we do see a not a significant, but a slight increase in the ratio compared to chronic GVHD, although not significant. And so, yeah, so just some other results to fill you in on. In the interest of time, I didn't want to put everything on here. But as I had mentioned before, we've had similar results across the majority of these assays with both knockout T-Regs or drug-treated T-Regs. All the drugs have pretty much worked pretty comparably as well. We also see significant increases in fatty acid uptake as well as fatty acid transporter expression with fatty acid synthesis inhibition. And so my initial hypothesis was that by limiting that malonyl-CoA production, we would be enhancing fatty acid oxidation. And this fatty acid oxidation would be increasing the OXPHOS, and the OXPHOS is what's increasing activity. All I've really measured up to this point is just OXPHOS, but there are other fuels that could be actually feeding into OXPHOS besides fatty acid oxidation. And so we still need to kind of nail down that it's fatty acid oxidation that's playing a key role in this. And then we've also successfully treated chronic GVHD mice with TOFA as well. So instead of going in with our knockout T-Regs at day 28, we started treating the mice with TOFA. And in this case, we actually saw a significant increase in T-Reg frequency and number in the spleen, which would be what we would expect based on that initial publication where they saw when you inhibit fatty acid synthesis during T conventional differentiation, you could actually drive differentiation toward T-Regs. And so that would be consistent with what I would expect. And then just to conclude, blocking fatty acid synthesis in mouse T-Regs results in increased OXPHOS and enhanced in vitro suppressive activity. This increased activity is correlated with an increase in inhibitory molecule expression as well as decreases in mTOR. And this fatty acid synthesis inhibition is also correlated with an increase in mitochondrial fusion, as well as an increase in mitochondrial activity. And the increase in T-Reg suppression is actually dependent on the increase in OXPHOS, as we're able to go in and inhibit both at the same time. And that leads to a loss of the enhanced suppression that we saw. And then chronic GVHD animals can be therapeutically treated by adoptively transferring our T-Regs. And then just some future directions. As I mentioned, we want to use 13 carbon labeling to really nail down what metabolic pathway is being utilized by these T-Regs. So the preliminary evidence suggests fatty acid oxidation, but we're interested in starting off testing glucose, glutamine, as well as fatty acid to see if those are all driving OXPHOS in these T-Regs. We still need to move these, try to assess this model in a GVL model, or try to assess targeting fatty acid synthesis in a GVL model. All we've done really is GVHD up to this point. And as I mentioned before, we're interested in leukemias and lymphomas. So eventually we need to kind of move into that kind of a model as well. Another interesting thing, or one thing that I've been wondering too, is how long does the benefit of fatty acid synthesis inhibition last in T-Reg? And is it ever detrimental? So, I mean, T cells need fatty acids, or not T cells, but just cells in general need fatty acid when they proliferate to divide and create more plasma membranes. And so there has to be a point where this could be detrimental, either during an expansion type of phase for T-Regs, or I don't know. So I'm just kind of interested to see if we could find evidence of that. And then we also want to just use RNA-seq to try to correlate some of these global gene changes that we've noted, or some of the enhanced metabolism and function with more global gene changes. And then just to acknowledge everyone in the Blazer lab. We've also worked closely with Amito Kelikar, Tim Sparwasser, and then Roddy O'Connor and Mike Dustin and Ewood over at Oxford. And that's it for me.
Thanks, Ethan. Great talk. It looks like we have a lot of immunologists in the crowd today. So yeah. You have a number of questions in the queue from Drs. Dame, Dr. Ruan, and Dr. Simmons. So I think you can read those. Maybe I'll let you go for it.
Yeah, so fatty acid synthase inhibitors, that's a great question. I have tested C75, and we do see, I haven't done a lot with it, but we do see a slight increase with that as well. Maybe even suggesting that malonyl-CoA isn't necessarily mediating the effects that I'm thinking, since basically fatty acid synthase is one step ahead of the ACC, and so you're getting that malonyl-CoA production. You're just not getting malonyl-CoA changed into whatever it's changed that to after that. So those are things that we're very interested in trying to test out right now. We've just started doing, or I did the C75 a while ago, and I've actually been meaning to pick it back up to try to relate it back to this. And then, can you rescue phenotypes by supplementing medium with lipids? So I've actually tried doing that. I've found that supplementing with exogenous palmitate is quite toxic to my cultures. So I haven't really found a good way to give the palmitate back. But yeah, that was something that I was trying to do. I don't know if anyone has any suggestions on culturing cells with palmitate and how to keep them viable, but you could email me or let me know after.
Ethan, be sure to read the whole question for people who aren't able to.
Oh yeah, sorry.
Just in case.
Sorry. Yeah, Scott was asking, can you rescue phenotypes by supplementing medium with lipids downstream of ACC, such as palmitate? And I tried that, but the palmitate was quite toxic, so it just killed my cells. and Ethan, did you provide exogenous free fatty acid in your seahorse OCR experiments? Normally there's little free fatty acid in culture medium. That is a great question. We did not, and that was one thing we've been trying to go to as well. There's a fatty acid oxidation seahorse assay where you add in exogenous fatty acid, and that's probably one of the next steps that I'm going to be doing. But I agree, there's very minimal free fatty acid in the culture media. There should be none, actually. So yeah. So if there is an increase in oxphos in my seahorse, it might not be due to the free fatty acids, because as you're suggesting, they're not there. Fatty acid synthesis inhibition depletes downstream lipid species. Did you look to identify lipid or lipid modified proteins that are necessary for T cell differentiation? We have not, and that is another thing that we're trying to do or want to do. All I've looked at as far as lipid composition has just been flow cytometry dyes, like the Bodippies. I've done Philippine, and then a collaborator in Oxford has done another dye that I can't remember the name of. We haven't really found one interesting thing, I guess, that we found, but isn't necessarily a strong point that I kind of want to add to my project yet, or add to the paper, is we see a slight, I don't know, I mean, a slight trend towards an increase in neutral lipid composition with fatty acid synthesis inhibition. And so that would be completely counter to what I would expect. I would expect that when we inhibit fatty acid synthesis, that we would lose, neutral lipids as well as some other lipids. But we actually tend to see a slight increase in neutral lipids. And so I don't know how that correlates yet. But yeah, so we have tried looking, but we also want to do some other more mass spec approaches to try to get a more comprehensive look at the lipid composition of our cells. And then finally, could you inhibit CPT1 simultaneously determine if the increase in OCR? Yep, and so that's actually again, another exact thing that we're going to be planning on doing. We're going to be using Etamoxir in combination with ND630 to see if the increase in suppression and oxphos is due to fatty acid oxidation specifically. But yeah, great questions.
Right. So Dr. Dickerson has a question, there's a question or two that's coming up in the chat rather than the questions. So Dr. Dickerson asks, since you are changing fatty acid synthesis, do you think adding an unsaturated fatty acid might be better, such as 18.1, 18.2, etc.?
I've actually never even considered that. So yeah, that's something I don't know. I don't know how adding unsaturated fatty acids to the system would affect it, or whether it would make it better. But that's definitely something I should look into.
And Dr. Pinnell, final question. Have you examined the B cell repertoire in these T-Reg recipients?
In the chronic GVHD, no, we have not. And we actually have a repeat of the in vivo chronic GVHD coming down in a month and a half or so, that we're going to be doing a lot more phenotyping in to try to get an idea of B cell phenotype, as well as a little bit more T cell phenotyping as far as cytokine production, as well as T-Reg phenotyping as well. So yeah, so our phenotyping right now in vivo is a little more limited than I would like, but we have plans to beef it up, certainly.
Wow, this is incredibly complex.
Carol, this is Sandy. It looks like we've got one person now with a hand raised, and maybe we could have this be our last question.
Yep.
All right.
Wrong. Oh, now the hand is gone.
Nope.
Not sure, they stepped out. Okay, we may be done then.
Glenn Simmons, final question. Maybe adding unsaturated fats along with palmitate to protect the cells. Aha, that's a great idea.
Yeah, that's actually a good idea. I've never, like I said, I've never even really thought about unsaturated fatty acids, so that's something I need to read up more on.
Okay, wow, you guys, this is awesome today. Thank you so much for your beautiful presentations and answers to questions. Very, very gorgeous work. And thank you all for staying on an extra 5 minutes. I think it was really successful. And have a great week, you guys.
Thank you.
Thank you, guys.